selamat pagi, everybody. Okay. Okay, shall we start? Shall we start? Can you see the slide? It's okay.
Yes, doctor. Yes, doctor.
Until time's done. Right. 9 a.m. Okay. How are you all? Are you all fine?
Yes, I'm fine.
Okay. Thank you. I feel something missing, you know, because I didn't see you. I can't see your face. So please reply your voice when I ask the questions, because I feel like something missing, you know. It's not real lectures, not
Okay,
classroom. Hopefully you can join the classroom, what you call the lectures, activities like practicals, tutorials soon in near future. Do you like the online teaching? Of course we have to, right? Okay. Can you see my picture, my photo? Can you see me?
Yes, doctor.
Okay. Okay. Clearly?
Yes.
Yes, doctor.
Can you see the PowerPoint slide I show right now?
Yes, doctor.
Okay, good. So very good. Okay, shall we start right now? My dear sisters and brothers, glad to be delivering lectures this morning. So I would like to provide the 2 hours lecture. In first hour, okay, I'm going to deliver about the introduction part of the hormones. So that once you gain the knowledge from this introduction part, you would appreciate about the, you know, the procedure, how to study the integrated course body systems. Actually, body systems arrange, you know, starting lectures are arranged in the body systems. Lectures are arranged from anatomy, physiology, biochemistry, and then later we move to the pathology and pharmacology later. And we also, what to call that, added the pharmacotherapeutics part, actually introduction part of the pharmacotherapeutics. So it is an integrated course, and then also the 3 credit hour course is a quite, what you call that, big courses. So as heavy as the cardiovascular system, what you studied last semester. Okay, without further ado, I would like to continue about the hormones. So as you have already known, hormones are the chemical messengers. So chemical messenger, they are secreted from the glands. So they are different from the exocrine glands, you know. Exocrine glands require ducts. So the enzymes are secreted from the exocrine glands, whereas endocrine glands secrete the hormones. So they are ductless glands, and then once hormones secreted from the endocrine part of endocrine glands, so hormones will travel to the bloodstream, and then later on bloodstream transported the hormones to the target cell, whereby the hormone exerts its physiological function after it passed through all the biochemical reactions and pathways, series of reactions. Series of reaction has to be done in order to perform the physiological functions well inside the target cell. Okay, my dear brothers and sisters. So you're going to study about the endocrine system and reproductive system. So the study is called endocrinology. So in endocrinology, we are going to study about the communication systems and the information transfer from the glands to the target tissue, and then you will be seeing there are 2 main regulatory systems in our body. So central nervous system and endocrine system, they have their own functions, but sometimes the central nervous system and endocrine system work together and regulate the cellular activities to maintain in a good health. Okay. So the nervous system is like telephone. If you want to call your mom or me, you just press the keyboard, and then the telephone you can directly call. So nervous system function is like electrical in nature and very fast. If you want to know something from me, you just ring me and you just message me, so I can directly reply. But endocrine system is a little bit different from nervous systems. It's like a radio broadcast. For example, let's say radio broadcast or some media, for example. So in order to know that your friend's current situation or friend's situation, you have to, switch on the Facebook, right? In the Facebook, some friends will upload the updates. So, and then also another example is the radio broadcast. So you have to switch on the radio broadcast. So you will be keep hearing about the updates about COVID. So you know the situation, which area is a red zone, which area is a yellow zone, so which area is already announced MCO. MCO is active in certain stage, something like that. So nervous systems and endocrine. So in that case, endocrine systems, in order for the hormone to exert its physiological function, it's require the receptor. So that's why in the next lecture, I will brief about the receptor before going to the, hormone action, mechanism of hormone action 1 and 2. Okay. So nervous systems and endocrine system is, as I mentioned earlier, they often work together to regulate the physiological functions. Okay, some neurons also have endocrine cell. For example, let's say hypothalamus have, there are some neurons function as endocrine system because they secrete the endocrine hormones. Okay, You will see later. So here you can see that neurosecretory cell, hypothalamus. So secrete the hormones to the either anterior and posterior part of the pituitary, chromophores, cells also what you call the secrete the hormones. That hormones are transported by the blood and enter into the either posterior and anterior part of the pituitary, and they stimulate the certain cells in posterior and anterior part of the pituitary to synthesize the hormone. So anterior pituitary also synthesize the hormones, various types of hormone that can control the other hormone synthesis. For example, let's say, okay, follicular stimulating and luteinizing hormone that stimulates the sex hormone synthesis in male's reproductive system and female reproductive system. Similar ways, ACTH that is synthesized from the anterior part of the pituitary, that is transported to, which is transported to the adrenal glands, in which adrenal hormones such as mineralocorticoids, glucocorticoids, and androgens are formed by the action and influence of the ACTH, something like that. So for the ACTH, secretion is also influenced by the neurosecretory cells. Hope you got it. Okay, so endocrine systems are present in our body, hypothalamus and then pituitary, pineal glands, and then at the neck, you can see that the thyroid, pairs of the thyroid gland, parathyroid gland. So when you check the, what you call the GI tract, gastrointestinal tract, at the pancreas is made up of with the exocrine part and endocrine parts. So endocrine parts of the pancreas are located in the islets or Langerhans. Isn't the islets of Langerhans also composed with the various types of cell, A cell, B cell, alpha cell? In other words, you can see that alpha cell, beta cell, gamma cell, delta cell. Can you hear me? Hello.
Yes, Doctor.
Okay, thank you. Oh, thank you. Thank you. Okay, then. So exocrine part, endocrine part secrete the insulin, glucagon, so on and so forth, right? So in order to regulate the blood glucose metabolism, fat metabolism, and protein metabolism. So again, in the reproductive, you can see that it's a kidney. Above the kidney, there are the adrenal glands or suprarenal glands. So suprarenal gland is the main, what you call that, the glands that synthesize the adrenocortical hormones like mineralocorticoids, glucocorticoids. So medullary part of the adrenal glands is important to synthesize the adrenaline, noradrenaline, and dopamine, so on and so forth. Female reproductive system is a site for synthesis of androgen and progesterone, so on and so forth. So male testicle also synthesize the testosterone and some male sex hormones and all that. So our endocrine system is scattered or in the body. Okay, my dear sisters and brothers. So endocrine system is very important, as you are aware, in a physiology, right? So endocrine function maintain the internal homeostasis. It supports the cell growth and differentiation. Endocrine system support us for to maintain the coordinate the development, coordinate the reproduction system, to facilitate the response to the external stimuli and internal stimuli. So when you check the parts of the endocrine systems, you can find that the 8, what you call the significant parts available in the endocrine systems. Number 1 is the sender. Sender meaning the sending cell where endocrine hormones are synthesized. So it's the mother glands. So that synthesize the hormone and send the signals is a hormone, chemical messenger. They are secreted out, excreted out into the bloodstream. So bloodstream is carry the hormones. So there are two main different types of the hormones. Some of the hormones are lipid soluble. Some of the hormones are water soluble protein in nature. So in that case, for the lipid soluble one, since they are unable to travel in the bloodstream, they require the transport protein. But protein hormones, since they are water soluble, they can travel simply in the bloodstream. So until they reach the target gland, target tissues. Okay, in the target tissue, they will bind to the receptor, as I mentioned earlier. So in order to show the secondary cellular response and physiological function in the target tissue, they require the receptors. Okay, so receptor is like a selected receiver that receive the message from the signals hormone. And later, once the receptor protein become active, the receptor protein will activate to the adjacent proteins. That protein is called transducer protein. So transducer protein transfer the hormone action to that protein. So activation of that transducer protein again activate to the another adjacent protein, maybe enzyme, and later on it will release the second messenger. So that carries the hormones action, and hormones, what it wants to do will be carried by the second messenger. So second messenger later on it's activate some innate enzyme inside the cell, and then make that enzyme become active. That enzyme will start to involve in the physiological functions. So you can find out. So in order for the hormones to show their function, it's required the receptors, transducer protein, amplifier proteins, effector proteins, and then later on, they will start to function so that we receive the what you call the specific action. For example, let's say insulin. You have already aware about the insulin. So insulin is secreted from the pancreas, and later insulin is transported to the liver and muscles, adipose tissue, so on and so forth, whereby insulin will bind to the receptor. So insulin binding to the receptor conformational changes happen, especially at the tyrosine amino acid residue. They the reaction modification is a phosphorylation reaction. So since phosphorylation reaction happen in tyrosine amino acid residues of the beta subunits of the insulin, so it will be activate to the adjacent protein enzyme, again also activate the other effectors protein, and finally, it will result the protein synthesis and glucose transporter synthesis, and then the gene activation, so on and so forth. Okay, so apart from the endocrine hormones in our body, also secrete the paracrine hormones and autocrine hormones. So what do you mean by paracrine hormones? For example, let's say like here you can see the paracrine hormones. So it arises from the cell and travel relatively small distance. It's different from our endocrine hormone. Endocrine hormone can travel from the pancreas to the whole body, something like that, the whole body. So paracrine is the adjacent cell. So they also require the receptors. For example, insulin-like growth factor, not insulin hormone. This is a specialized growth factor involved in the growth and differentiations of the cell. So secreted by the muscle cell, may promote the growth of neighboring muscle cell and connective tissue. So it's an example of paracrine hormones. So another hormones, what we can find in our bodies are the autocrine hormones. So they are produced in the same cell, autocrine same cell, the cell that synthesizes the hormone, paracrine hormones, is also target cell. For example, let's say when you mosquito bite to your skin. So your skin later on will become red, swelling, right? Because of the the intracellular synthesis of histamine and prostaglandins, you know. So cell becomes swollen and then swollen because of edema, and then the change to the red color. And also you feel pain and itchy because histamine, prostaglandins are released from that part. So that's why now you know that apart from the endocrine hormones, we have some other hormones, paracrine hormones and autocrine hormones. Okay, so as I mentioned earlier, my dear sisters and brothers, we will have mainly two types of hormone. Some of the hormones, steroid in nature, they are derived from the cholesterol. They are fat soluble. So when you check it, you will find that all the adrenocortical hormones, sex hormone, dihydroxycholecalciferol, and vitamin A, retinol. So they all are the fat soluble. So in order for them to be transported to the target tissue, they require the transporter protein. So water-soluble protein like peptides and protein, the common examples are, as you already know, insulin, parathyroid hormone, ADH, glucagon, so on and so forth. There are many hormones which are synthesized from the anterior part of the pituitaries as well. They all are protein and peptide hormone. What is different from protein and peptide? You know, right? If peptide change, amino acid sequence is more than 51 is considered, 50 is considered protein hormone. The hormone, the amino acid sequence change is shorter than the 50 is called the peptide hormone. This is a different terminology how to term the peptides and protein hormone. Okay, my dear sisters and brothers, now you can see that this is a structure of cholesterol, right? You have already remembered since your first year, second year. So cholesterol structure, cholesterol is the main precursor to synthesize the different steroids hormone, and they have different important role in our body. Okay, my dear sister. Another type is a protein, but it's attached with the glucose moiety. So glucose moiety is very important in order to stabilize the hormone structure. For example, thyroid stimulating hormone that is synthesized from the anterior part of the pituitary, follicle stimulating hormone, luteinizing hormone, human chorionic gonadotropin hormone, which is synthesized in the placenta during the pregnancy. So these are the glycoprotein in nature. So another type of hormones I would like to introduce you is that amino acid derivatives hormone. So that means, catecholamines, thyroid hormones like triiodothyronine, thyroxine, they are derived from the amino acid tyrosine. You can see the structure here. So tyrosine is the precursor amino acid that can transform the catecholamines in adrenal medulla. In thyroids, it can form the thyroid hormones. But depend on the structure, since catecholamine in the final stage, it's already hydroxylated, it's become the water soluble. So in order for them to exert the secondary cellular response, they will follow the procedure like protein hormone. But thyroid hormones, in the final process, and then the thyroid hormones become a fat soluble. Depend on their structures. I will show you the structure detail so later. So the structure make the thyroid hormone become fat soluble. For the fat soluble thyroid hormones, in order for them to show its physiological function in target cell, it doesn't require the membranous receptors. What require is that it will require the intracellular receptors only. Okay, so there are some other hormones like prostaglandins, arachidonic acid is also what you call the autocrine hormone, right? So autocrine hormone is derived from the lipids. Do you know that? Where are they derived? They are derived from the essential fatty acid, right? And omega-3 fatty acid, omega-6 fatty acid. So these are the major precursor for the prostaglandins and arachidonic acid. They have what you call that they have hormone activities, local hormones activities. Okay, my dear sisters and brother, but we have various types of the now you have already know that various types of hormones, but depend on the what is solubility. So we would just classify to the two hormones into the two types, two groups only. For example, steroid family, they are lipophilic and hydrophobic. They require the transport protein. So plasma half-life is quite long. And then the receptor, they do not need the, can you hear me, brothers, sisters? Hello?
Yes, sir.
Okay, thank you. So they can cross the plasma membrane because plasma membrane are lipid bilayer. They can cross the plasma membrane, but they also require the receptor in order to bind the specific DNA region. So they require intracellular receptors. Later on, intracellular receptor and hormone binding will change to the hormone receptor complex. The hormone receptor complex will directly bind to the DNA binding region and will initiate the transcription process and then translation process, and then later on, as a result, specific proteins will be formed. That protein will function on behalf of the steroid hormones. Okay, so when we look at about the peptide and protein and glycoprotein hormone, amino acid derivatives hormone, so they are the hydrophilic in nature. So they do not need any transport protein for the transfer and travel in the bloodstream. So since they are polypeptide and protein, once they function the cellular activities, and then they will be degraded by the specific peptides, specific proteases. So that's why their plasma half-life is. And then, of course, since they cannot cross the cell membrane, they require the plasma membrane receptors. Okay, under this class, you can find that they will hire the second messenger inside the cell in the target tissue. So depend on the types of the second messenger, the hormone, specific hormone hire, specific hormone use. So we will have various type of subclass in the group 2 as well. Cyclic AMP, cyclic GMP, calcium, diacylglycerol, inositol-phosphate, and kinase cascade. These are the different second messenger will be used by the different hormone in order for them to show physiological functions. Okay, my dear sisters and brothers. So signaling pathway. So signaling pathway is very important. So the stimulus may originate from the external or internal. So for example, let's say pain, pain stimuli. So when mosquito bite your skin, you feel pain. So that signal will send to the higher center, right? The brain. So you know that you are aware that mosquito has bitten you. Okay, so similar ways go to that. For example, I would like to give you the example that is the insulin. So what's the stimuli for insulin release? My dear sisters and brothers, what are the main stimuli that stimulate the beta cells of pancreas to stimulate the insulin release?
Glucose. Level of glucose.
And
Because of high level, then pancreas
Blood
detect.
glucose, yes. After eating, once the blood glucose concentration is high in the blood, that send the signals to the, what you call that, signal to the beta cells, right? So that beta cells secrete the insulin in order to lower down and maintain the blood glucose level in the plasma, right? Okay, so there some of the hormones are not secreted at the constant rate. It's depend on the stimuli. So their secretion is regulated by 3 different methods. You may see that. Action of the substance other than the hormone. So this is, as I mentioned earlier, glucose is the good example, stimulate the hormone secretions. So neural control of the endocrine glands. The example is the GI tract hormones. It's a control by the vagus nerve, isn't it? So control of the secretory activity of the one endocrine glands by hormones or neurohormones secreted by another endocrine glands. So that means, for example, thyroid stimulating hormone secreted from the anterior part of the pituitary that stimulate the thyroid to synthesize the thyroxine, triiodothyronine, so on and so forth. That is we call hypothalamo-pituitary axis. So that, what you call that, signaling pathway is very important for the hormone secretion, hormone regulations as well. So you can see that. So we can see that some of the hormones follows the regulations. So positive feedback regulation, and there is a negative feedback regulation. For example, let's say it's more or less similar like enzymatic activities. For example, let's say, so in order for the enzyme to initiate the reaction, enzyme must have a substrate, right? So substrate enzyme bind, and then also produce the product. So as long as substrate is available, enzyme is available, it will produce the product. But in certain time, so the product is formed sufficient amount, adequate amount, the product will inhibit the action of enzyme. So finally reaction stop, isn't it? Similar ways go to the hormone regulation negative feedback. For example, let's say your luteinizing hormone and follicular stimulating hormone level is really high before ovulation. So because they are what you call that, they are what you call that, stimulate to the your Graafian follicles, what you call that, the growth, and then also estrogen and progesterone synthesis. Once hormones start to form, so they will send the signal to the anterior part of the pituitary from the ovaries. So what you call that, requests to stop the luteinizing hormone, follicular stimulating hormone, so on and so forth. So finally, it's the product formation and what you call that, target glands hormones formation will try to stop the stimulating hormone synthesis from the other glands. Okay. Okay, and then also hormones have positive feedback regulation, of course. Before ovulation, estrogen, there is no estrogen and progesterone form. So at that time, no ovum is not formed yet. So that's why in that before ovulation time, so there is no product, right? In that case, the low concentration of the hormone, very less amount of the hormone, the ovaries send the signal to the anterior part of pituitary to synthesize more follicle stimulating hormone and luteinizing hormone, so on and so forth. So that's why our body is intricate, very intricate. They are strictly control each other and efficiently control each other. For example, hypothalamus control to the pituitary control to the other endocrine glands, what you call the systematically. Okay, you can see that. Okay, here, positive feedback. Let's see here. Okay, before ovulation, so ovary send the signal, estrogen, less amount of estrogen send the signal to the hypothalamus. So hypothalamus, what you call that, recognize the stimuli and start to form the gonadotropin releasing hormone much. So increased amount of gonadotrophin releasing hormone also again send the signal or stimulate the anterior part of the pituitary to synthesize more luteinizing hormone. So luteinizing hormones release and transported to the ovary, whereby ovaries start to form the estrogen more and more and more, and also ovum to develop the ovum. So once what you call that, ovulations happen, what happen is that progesterone is released. The progesterone will send the negative feedback to the higher center, hypothalamus. So gonadotrophin releasing hormone synthesis stop. There is no stimuli to the luteinizing hormone. Okay, luteinizing hormone, finally luteinizing hormone or follicular stimulating hormone synthesis will be inhibited. Okay, decline. Okay, another example here, my dear sisters and brother, can you have a look? Hypothalamus, there is a thiotrope cell. Thiotrope cell release the thyrotrophin releasing hormone. Thyrotrophin releasing hormone stimulate the anterior part of the pituitary to synthesize a thyroid stimulating cell. So thyroid stimulating hormones. So hormones will also again stimulate its target gland thyroid so that T3, T4 are formed. So when T3 and T4 are formed in adequate amount, sufficient amount, that we send the signal to the anterior part of the pituitary and then also to the hypothalamus so that it stops the hormone secretion. Another one is a short peptide. Short peptide is also synthesized from the hypothalamus, and it has inhibitory activity to the pituitary so that the SH secretion may be temporarily stopped. So that's why hormone secretion is sometimes it's on, switch on hormone synthesis, sometimes hormone synthesis switch off, depends on the requirement, body's demands, okay? And another example, can you see here? In the male sex glands testes, luteinizing hormone activate the Leydig cell. So Leydig cells synthesize the testosterone. So testosterone assists to the sperm to form the mature sperm, spermatogenesis. Okay. On the other hand, follicle stimulating hormone stimulate the Sertoli cell, other cells which are available in the testes. So they also activate the spermatogenesis, work together with the testosterone. At the same time, Sertoli cells secrete the inhibin, other type of the protein which has the ability to inhibit the release of follicle stimulating hormone once the the testes require the efficient sperm formation. Okay, so also the similar ways, and then so testosterone is formed in sufficient amount, adequate amount. They do not need the extra hormones, so they will send the signal either to the anterior part of the pituitary and hypothalamus to stop the hormone secretion for a while. So that is how they are communicating each other. So very nice, right? So if we don't need, the demand is declined, and then so its demand is higher, the production will be higher from the higher center. We do not need a demand because we have adequate amount of amount of the product, so we don't need any supply, so supply stop. Okay. Okay, hormone secretion is changed time by time. So I would like to share you this one. Okay, so chronic hormone regulation, that means that thyroid hormone, so it's regulation, thyroid hormone is maintained in our bodies for quite long times, days and weeks. So acute hormone, for example, let's say a good example is stress. If you are challenging with the stress, like, especially during this time, we are facing with the new pandemic disease, we're really scared, and then of course, as a response, the stress hormone will be released, you know, in elevated level in our body. So that's why you can see that epinephrine, cortisol level. So they are acute regulation. When the stimuli come, hormone is secreted in higher amount. So some of the hormones are following the cyclical regulation. For example, female reproductive hormones, and then the luteinizing hormone, follicular stimulating hormone. So we have an ovulation period, right? Before 14 days, LH and what you call that, follicular stimulating hormone level will be elevated in the plasma. After the ovulation, 14 days after the menstrual period, and then estrogen and progesterone will be gradually high. Okay, so you can see that changes of hormone secretion time by time. Okay, so we'd like to share you the biosynthesis of hormones. Okay, so biosynthesis of hormones, since hormones are different, it depends on their structure. So their biosynthesis will be also different, definitely. So for example, let's see the protein hormone. So protein hormones, example is the thyroid insulin, and then the parathyroid hormone. So when they are formed under the guidance of gene expression, could you imagine that? So insulin is the pre-pro-insulin, very long chain peptide, right? So in the endoplasmic reticulum of the beta cell, the signal peptidase trim the amino terminus part of the pre-pro-insulin, so change will be shorter to form the pro-insulin. So pro-insulin is A change of the peptide and B change of peptide connected by the connecting peptide. Then it's also trimmed in the Golgi apparatus to form the mature insulin. So in the mature insulin, only contain the A chain and B chain, and total amount of amino acids that form the insulins are only 51 amino acids. Pre-pro-insulin, very long chain, you know, and even pro-insulin contain 84 amino acids in the structure. So they all are trimmed out and finally form the insulin. So that's why the different hormones have a different, what you call the biosynthesis process. And then parathyroid hormone, also like insulin, once they are formed, very long chain of the amino acid chain, peptide chain, and in order for them to reach the maturity, matured, what you call their functional hormones. So some of the unnecessary part of the amino acid sequence will be processed. Okay. Okay, so another one example I would like to explain you is that pro-opiomelanocortin. So that is a gene present in the specific chromosome, and then that what you call that synthesize the pro-opiomelanocortin polypeptide. Once they are formed, they have 241 amino acid residue, very long chain. And then later on, okay, actually, when they are synthesized in the anterior part of the pituitary, you know, they contain 285 amino acid long polypeptide precursor. Later on, they are trimmed, especially at the peptide part, 44 amino acid sequence from the amino terminus part of the long protein chain. So the chain becomes shorter, and interestingly, you know, you can find this kind of situation happen in anterior part of the pituitary. Okay, so pro-opiomelanocortin protein are trimmed, right? Signal peptide is removed from the POMC, become active. So activated POMC also again, the peptide from the carboxy terminus part will transform to the beta lipotrophic hormone, and the rest part that remain in the amino terminus part will form to the pro-ACTH. Later on, pro-ACTH trim two parts. The pro-ACTH is divided into the two parts, and then the second part will be the ACTH, mature ACTH, and then the rest part will change to the another peptide. It's about 16 kilodalton weight. Okay, so ACTH again, also ACTH is transported to the adrenal glands, right? Again, in the sum of the ACTH can be trimmed, peptide chains are trimmed to form CLIP, a melanocyte stimulating hormone. Okay, so in that case, CLIP is corticotrophin-like intermediate low peptide. So there are peptides. So various types of peptides are originated from the pro-opiomelanocortin gene. So actually that gene is present in the chromosome number 2, beyond 23 to 0.3 area of the gene, functional gene. How interesting, okay? So gamma melanocytes are also derived from the pro-opiomelanocortin, and alpha, beta melanocyte stimulating, endorphin, you can see the endorphin, lipotrophin, gamma lipotrophin hormones also are derived from the one peptide. That peptide is called pro-opiomelanocortin, very long chain peptide. Once it's trimmed, it will produces the various types of hormones. Okay, this is how ACTH are synthesized in the anterior part of the pituitary. Okay, my dear sisters and brothers, let me continue about the biosynthesis. Okay, that is about the T3 and T4. Okay, so in the endocrine glands, thyroid gland synthesize the D4 and D3, from the tyrosine amino acid, right? Okay, so later T4 is can combat the T3 triiodothione in the liver because T3 is the most active form. We can study its physiological function. T4 doesn't have this function. So we can say T4 is the inactive form of the thyroid hormone, but T3 is the active. So activations occur in the target tissue. Okay, occur in the target tissue because you know why? Thyroid mother glands synthesize only 20% of total population of thyroid hormone. So that's why in order for the T4 to change the T3, they are transported to the liver. And then later, and they are transported to the peripheral tissue. They play important role in growth, body growth, and maintain the thermoregulations, thermogenesis function later on. Okay, so that's why, some hormones have their specific, characteristics in terms of synthesis, in terms of transport, in terms of metabolism. Okay, so we'd like to explain about the DHEA. So DHEA is the prohormone. So dihydroxyepinandrosterol, this is the intermediate hormones that is derived from the cholesterol. So what happened inside the adrenal cortex, DHEA is formed before they are converted to the androgen. So half of them are transported to the liver, so in which they undergo the attachment of sulfate group. There is a sulfation reaction. That is the inactive form of the DHEA. So once your body require more androgen, the DHEA will transport it back to the adrenal cortex and then the sex glands, whereby, they will transform to the androgen and then the androgen and sex glands. Okay, so DHEA. So for the DHEA, it's transported to the liver, but actually liver is not a target tissue. It's a temporary storage organ. So that's why in the formation of androgen, so the certain step, okay, the activation reaction occur in the liver. So it's a very interesting step one. Okay, okay, okay. This last part. Naturally in the adrenal cortex, sufficient reaction, not only in the liver. During the first part, metabolism and liver and intestine, when exogenous DHEA is administered orally. Okay, nowadays the couple, they do not have a baby for many years. They are advised to take the DHEA supplement. So in that case, so once they consume the DHEA are absorbed in intestine, and then later on they are transported to the liver, stored for a while, and later they are transported to the adrenal glands and sex glands in order to synthesize the sex hormone biosynthesis. Okay, okay, another one. Vitamin D, actually, vitamin D, do you know that vitamin D is very important and popular nowadays? You know, some research observed that the most of the COVID victims are found vitamin D deficiency. So that's why scientists, you know, groups of scientists suggested that to take the vitamin D3 supplement as many as possible, because the vitamin D receptor will, what you call that, replace the binding of virus into the cells. So this finding is very important finding nowadays. So what I would like to share you is that vitamin D is very interesting. Actually, vitamin D is mainly and freely what you call the received from the sunrise. You know, when our skin exposed with the sun rays, so under the what you call the dermis, there is a fat layer, right? Cholesterol. 7-dehydrocholesterol will what you call that is transported to the liver, and also it will directly convert to the vitamin D3 and cholecalciferol, and also in the liver they are transported to the liver, 7-dehydrocholesterol, and then 25-hydroxylate make hydroxylation reaction at the ring structure of the cholesterol and make it 25-hydroxycholecalciferol, and later they are transported to the kidney, whereby kidney continue the next hydroxylation reaction at number 1 and number 24. So 24-25 hydroxycholecalciferol and 125 dihydroxycholecalciferol. Among the various type of vitamin D3, the 125 dihydroxycholecalciferol is the biologically active form, and they can initiate, they can show the physiological function. So when you look at the picture, you will realize that, you know, liver is not the target tissue, but 7-dehydrocholesterol must be transported to the liver to receive the first hydroxylation reaction. And later on, they are transported to its target tissue and to continue the, to complete the other hydroxylation reaction in order to change the most active form, biochemically active form. So in the liver, they do not have, vitamin D do not have any actions, any physiological action, but the kidney has. So renal tubular cells, in the renal tubular cell, so vitamin C play important role in the calcium absorption, calcium reabsorption, and inhibit the calcium excretion so that plasma calcium level is maintained high. So plasma calcium is required for the bone growth and development, and calcium is important for the blood clotting, and you will the importance of the calcium and hormone signalings. So calcium play important role as a second messenger for the certain hormones. So that's why you can see that biosynthesis of the calcitriol 125 dihydroxycholecalciferol in other name. It's require this kind of process, you know, this kind of process. So they have enzymatic modification occur in the non-target tissue, and they also require the enzymatic modification in the target tissue. Later, the mature form is formed, and they play important role in calcium homeostasis. So you can see there. And then this is the slide. Have you got it, my dear sisters and brother? Any questions so far? Any questions so far? Are you sleeping now?
No.
No,
You're here,
doctor.
Doctor.
Because I observe that, you know, my sons, you know, also study the online class. While he's studying online class, he lying on the bed. He lying on the bed and then, you know, comfortably lying down and then listening what teachers say. Okay, anyway, okay. So transport of hormone, as I mentioned recently, the steroid hormone require the transport protein. Example, though T3 and T4, they are derivatives of amino acid in terms of their structure, they are hydrophobic. So in order for them to travel in the bloodstream, they require the transporter protein, albumin, and globulin. So we call thyroid binding globulin. We can measure the amount in the plasma. So TBT normal T4, usually 99.98% of T4 bind to the globulin. The rest only 0.2% bind to the albumin. Okay, and sometimes you can find the free form. Very little amount will release out. Can be detected in the plasma, a very small amount. How about the other steroid hormone? The glucocorticoids bind to the globulin. So once globulin bind to the glucocorticoids, we name this what you call the glucocorticoids globulin complex is transcortin or corticosteroid binding protein. Of course, the testosterone, estrogen, since they are lipid soluble in nature, they require the globulin. So we can call sex hormone binding globulin. We can name sex hormone binding globulin when they are transported from the sex glands to the peripheral tissue. Because you know why estrogen is required for the breast development, so they are transported from the ovaries to the what you call that, to the breast. And then for the men also, in order for men to have secondary sex characteristics like hair growth and all that, so the testosterone are transported from the testes to the other peripheral tissues area. Okay, you can see that. Okay, concentrations of hormone. We have actually very minute amount of the hormones are secreted from their target cell, but the action is very powerful. You can see that the hormones ranges from the 10 to the power minus 15 to 10 to the power minus 9 mole per liter. So there are very tiny amounts secreted in our body, but they can function well in order to maintain our health. Okay, some hormone level are up to the 10 to the power minus 5 to 10 to the power 3. Depend on the physiological abbreviation. Very simple. So when you detect your adrenaline level, when you do not have any stress, I mean the stress-free time, your plasma level or adrenaline will very minimal amount will be detected. So once you got a stressful, you are encountering with the challenging situation. So when we withdraw the blood at that time and then check the plasma adrenaline level, definitely they are elevated, something like that. So depend on the situation. Similar ways go to the insulin as well. Plasma insulin level found very minimum in the early in the morning fasting time. So, but the level will be increased for 10 times, 10 folds times higher than the baseline data when you finish your meal and after eating your carbohydrate-rich meal, so on and so forth. Okay, so steroid except thyroid hormones can be stored. So there are the differences. Few hours for some water-soluble hormones, as I mentioned earlier. So water-soluble hormone lifespan is very short. Once they perform the action, they will be degraded by their specific proteases. For example, once insulin exerts its physiological function, insulin protein are degraded by the insulinase enzymes, peptides. Okay, so simple, right? Okay, the steroid hormones are different from the protein breakdown because steroid hormone are lipid soluble. So in order for the steroid hormone, catabolism is totally different from the protein hormones. So steroid hormones, when they perform the function, so they are what to call that? They have to undergo the enzymatic modification. First step is hydroxylation reaction and desaturation at the unsaturated bonds in the structure. And later on, they undergo the conjugation reaction. So conjugation reaction require the conjugation agents. They are the conjugants. Conjugants, they are glucuronide and sulfate, sulfation reaction, phosphorylation reaction. So after that, the steroid hormone change to the structures. So the conjugated form are water soluble a bit, so they can be excreted in the urine. So only 20 to 30% of the metabolized, I mean the conjugated forms, are secreted in the bile. Later, the ones bile are secreted into the GI tract, they are ultimately removed from the body by feces. Okay, the remainder is the conjugated form are fairly water soluble. They can be excreted through urine. Okay, my dear sisters and brother. Okay, so you can see that here is a normal reference value of the plasma hormone level for your information. So ACTH early in the morning, the amount in the blood is 10 to 18 nanogram per liter. Aldosterone level in normal, cortisol level is a high early in the morning. Gradually, once they function actively to maintain our health throughout the day, the midnight, the hormone secretion is lower than the 100 nanomole per liter. The follicular stimulating hormones in the ladies, okay? And postmenopausal follicular stimulating hormones level is higher than the reproductive, that amount of reproductive age group. You know why? Because the postmenopause, and then they do not have enough estrogen and progesterone. So that's why product is very less amount. So they will send a signal to the positive feedback regulation, which goes up to the anterior pituitary. So the follicle stimulating hormone secretion is high in postmenopausal growth hormone. Okay, the glucose load will be the higher. So insulin in fasting is this amount. And later after meal will be higher luteinizing hormone in male and female, also similar weights like follicle stimulating hormone. Postmenopausal women, so luteinizing hormone level is high. You can see parathyroid hormone, prolactin hormone that is secreted in higher amount in the lactating mothers, and some other tumors, you know, prolactin releasing tumors. We call it prolactinoma. It's the anterior pituitary has what you call the tumor mass, and then in which whereby the prolactin is release in excessive amount. Okay, so that one pathology we explain detail. Okay, okay, testosterone amount in a female, because you know why? Before that, you simply know that, oh, testosterone is male hormone, androgen. I mean, the estrogen is a female sex hormone, but you will know detail about that. Both female and male, we do have both testosterone and estrogen. But male, of course, testosterone dominating the estrogen. Okay, the amount is higher. Okay, so similar ways go to the female. Female has high amount of estrogen, but at the same time, female also do have a very small amount of testosterone, so which can be synthesized from the your adrenal cortex, androgen from adrenal cortex. So how we are created, you know, very interesting. Okay, direct stimulating hormone amount, T3 and T4, total and free. And free androgen index is we need to know when you study about the biosynthesis of the sex hormones. Okay. Okay, my dear sisters and brother, it's I think last slide for this lecture. So very interesting. So these are the some other factors influence the hormone production. So hormone production is a variable. For example, as I mentioned, as you have already seen, because the cortisol level is found highest in early in the morning, gradually decline until, you know, you will find the least amount of cortisol, serum cortisol in the midnight. So they follow the diurnal variation. So time of the month, as I mentioned earlier, before the menstrual cycle, LH, FSH level is high. After ovulation, estradiol level will be so on and so forth. And melatonin. So melatonin is also nowadays very popular supplement for the COVID patients, and then those who for the prevention of the COVID-19 infections as well, like a vitamin D3. So some researcher and medical practitioners suggested to take the vitamin D3 supplement, vitamin C, zinc together with the melatonin. So the melatonin hormones level are found higher in winter period where weather is cold, and then you can see that T3 level are lower in the elderly subject. Of course, the age is a very determining factors and influence the hormone production. For example, let's say my dear sisters and brother, your hormone secretion is higher than me because I'm a 55 and quite old. My hormones, you know, the glands are gradually filled to secrete the adequate amount of all hormones, including T3, T4, sex steroids, and I mean estrogen, progesterone. Even my estrogen and progesterone hormone secretion is already stopped. Okay, just sharing. Okay, the sex hormone, of course, men, you have a higher amount of testosterone and very least amount of estradiol. Estradiol means E2, estrogen, the same name. And then but female, the estradiol, estrone level is higher than the testosterone. Okay, stress. Of course, during the stress, your cortisol and adrenaline or adrenaline level are very high. This is a very beautiful questions, you know, usually asked in short notes. Okay, please take note that. And the diet, of course, insulin level is high when plasma glucose is high. Growth hormone is also when glucose load is high, the growth hormone will be suppressed. Okay, so cortisol change, okay, and then illness, of course, non-thyroid illness, like for example, as let's say you have a high fever, maybe due to the bacterial infection, viral infection, whatever infection comes, it resulting in a high fever, it will alter your thyroid function test. So you will find that the false positively high level of the thyroid hormones. And certain drugs also can influence the thyroid function test, like amiodarone and contraceptive pills that also alter the thyroid function test. So that's all about my introduction lecture, my dear sisters and brothers. So shall we stop for a while? Shall we stop for a while?
Yes, doctor.
How many minutes you want me to stop?
Yes, doctor.
How many minutes you want me? 5 or 10 minutes?
10.
10 minutes, okay. So take rest for a while. I won't go out. I will leave like that. Okay. You can take rest for a while.
Okay,
Take
thank you, doctor.
Thank you, doctor.
rest for 10 minutes. I will come back. Okay. So any questions who want to ask any questions from anybody? Any questions? Would you like to ask any question? Reach me anytime. Okay. Okay. I don't leave. I will.
Doctor,
Yes, sister.
can you
Yes.
repeat the positive feedback mechanism?
Oh, positive feedback. You mean? Is it?
Yes.
Okay.
Yes.
Is it Hajar?
Yes, doctor.
I remember your voice. Okay, so positive feedback meaning to say that okay, so I give you the example about the enzyme regulation, right? Once enzyme in a metabolism, if you do not have a product yet, for example, if you body demands is high for the glucose, let's say. So glucose is the concentration is lower during fasting time. So the glucose will be produced from the non-other glucose sources like amino acid, right? So in that case, what to call that? The gluconeogenic, the enzyme involved in key enzyme involved in gluconeogenesis will become very active. And then active try to produce the glucose. Once the glucose is formed, and then when you break the fasting, and then you try to eat the food, carbohydrate-rich food, your blood sugar level is rise up. In that case, the key enzyme activity is automatically switched off. Okay, similar ways goes to the your what to call that, your hormones regulation as well. For example, let's say, okay, so body require adequate amount of T3 and T4 for the growth and development to maintain our thermogenic function, calorigenic function, and all that, right? If we have adequate amount of, our body is actually very good sensor, we have adequate amount of T3 and T4. We don't have, let's say, at the beginning, positive, right? You are asking about positive. So if we don't have adequate amount of T3 and T4 yet, or similar meaning is that T3 and T4 level are found very low. So that means hypothyroid condition, right? Hypothyroid, let's see. So thyroid hormone is unable to secrete the hormone in adequate amount. In that case, since your hormones level from the endocrine glands is low, that we send a signal to the pituitary. Okay, pituitary. So pituitary gland synthesize the thyroid stimulating hormone system. So thyroid stimulating hormone will be synthesized more than previous time. So thyroid stimulating hormone synthesize more and try to stimulate the thyroid glands to synthesize more and more and more T3 and T4 because T3 and T4 demand is high in your body. So that positive regulation can increase the T3 and T4 level as a result of positive feedback regulation. Got it, Hajar?
So it means it keeping increasing and increasing.
Yes, increasing until it's reached the optimal level.
Okay.
Okay,
Yes.
so I would like to share a few more information since you are very interested. Okay, so we have two pathological conditions. Actually, Dr. Sanda will explain the pathology later on after my lectures. Okay, so hypothyroid is a disease in which your T3, T4 concentration is low because of some antibodies attack to the thyroid tissues. Okay, so in that case, thyroid T3 and T4 synthesis is very low. So send the signal to the, of course, this condition resulting sending the signal to the pituitary. Anterior part of the pituitary, so synthesize more TSH, more TSH. So TSH try to stimulate the thyroid glands to synthesize more and more thyroid T3 and T4. But since pathology happen inside the thyroid glands, so T3 and T4 level wouldn't be increased like normal person. So in that case, in order to confirm the diagnosis of hypothyroidism, so we have to check the both T3 and T4 and TSH plasma level of TSH, very important. Definitely you will find the low T3 and T4, serum T3 and T4 level together with the increase the TSH level. So you can make the confirmation of the what you call that diagnosis. So this is very important for us. This knowledge we gain from the, you know, the biochemistry, you can apply this knowledge in the not only in the pathology but also in the what you call the treatment, clinical and pharmacotherapeutics as well. So hope you get it, Sha, Haja.
Get it, Doctor.
Okay,
Thank you.
in a hypothyroid also, hypothyroid means the condition whereby thyroid cell, what you call the stimulate the more T3 and T4 formation because of maybe there's some goiter, you know, goiter, Graves' disease and all that. So these are the condition in which the thyroid hormone are secreted in exaggerated amount, aggressive amount. So thyroid hormones is high. So in that case, so high level of thyroid hormone send the negative feedback to the pituitary gland. So pituitary glands are unable to synthesize the TSH. Finally, TSH wouldn't stimulate the thyroid. So in normal, once TSH is activity is switched off, no thyroid hormone biosynthesis. But in case of Graves' disease, the T3 and T4 is keep secreting from the thyroid gland. So in order to confirm the hyperthyroid, we have to check the thyroid function tests that include the T3, T4 level and TSH level. In hyperthyroid cases, you will find high level, elevated level of serum T3 and T4, but very low amount of TSH. So that is an indicator to confirm the diagnosis of hyperthyroid. It is a what you call that directly. It is a reverse from the hypothyroid cases. Okay. Okay, Ja. Any questions, sisters, brothers? Any questions? Any questions? Can we restart again? Hello?
Yes, doctor.
Can you see the slide, PowerPoint slide I shared?
Yes, you can see it, doctor.
Okay, sure. So shall we go ahead? So it's a time to talk about the hormone actions. So we can classify the hormone, right? To class group 1 and group 2. So the group 1 belongs to the hormone that binds to the intracellular receptor before they are showing the cellular functions. And the proteins hormone, and it's a group 2 hormone. So they cannot cross the cell membrane of the target tissue. So they are require the membranous protein. They are required to bind the membranous protein. Later, they have to hire the specific second messenger. Hormone is the first messenger. So the second messenger will carry the information that give from the hormone, that is given from the hormone by the hormone to the cell target tissue. So that's why, for example, ACTH, antidiuretic hormone, parathyroid hormone, follicle stimulating hormone, luteinizing hormone, and or what you call the cortical releasing hormone. So these hormones are under the subclass A, group 2A, because they use that cyclic AMP. They hire the intracellular cyclic AMP to show the physiological function in the target tissue. So atrial peptide, and then the natriuretic peptide, and then the nitric oxide are they use the cyclic GMP to exert their physiological function inside in their target tissue. So there are a group of hormones, the acetylcholine, alpha-adrenergic hormones, and then the thyrotrophin releasing hormone. They use that other type of second messenger, diacylglycerol, phosphoinositol, and calcium. Okay, for them to show their physiological function. And insulin like growth factor, nerve growth factor, epidermal growth factor. There are so many factors because insulin is also considered anabolic hormone, right? Insulin can increase all the synthesis like protein synthesis, fat synthesis, and then the protein synthesis. There are so many growth factor synthesis require insulin. So insulin higher the kinase or phosphatase cascade. So it is receptor binding will cause the kinase and phosphatase cascade. And then later on, they will show their physiological function. Before going detail to their mechanism of action, would like to revise about the receptor. I think you have already known. So receptor may have intracellular receptor or membranous receptor. They are glycoprotein in nature. So they have, since we have two types of receptor, some of the receptors are present inside the cell, some are located on the membrane. Okay, they have three functional domains. Number one is recognition domain. So that part recognize the incoming hormone, so they will accept the hormone. So hormone binding domain or ligand binding domain, and the receptor is exposing outside and also embedded inside in the plasma membrane. So that's why they have transmembrane domain. And also the last part, there is a catalytic domain or signal transduction domain. They carry the signal from the first messenger hormone, and later they will bind to the other enzyme. So they have a catalytic domain that provide the reaction with the intracellular enzyme. So 2 and 3 are domain that generates a signal. Later, they will alter the intracellular functions. So cell surface receptor. Cell surface receptor is important for the water-soluble hormone. They will generate the cascade of intracellular signals and alter the behavior of the cells. So there are various types of membrane receptors you can see. So ion channel link receptor, they allow the ion, so sodium, potassium, chloride ion to go in and out of the cell, okay? So they can change the permeability of the plasma membrane and the excitability of the postsynaptic cell. So example is that intracellular, sodium level will, important for the insulin hormones excretion from the endocrine glands. Okay, there are another type of receptor. There is a G protein linked receptor. The receptor and hormone binding to the receptor linked to the G protein initiate the activate the G protein, so that activated G protein will proceed the other functions. So enzyme linked receptor. So the enzyme linked receptor, in that case, the receptor itself is an enzyme, you know, it's function as an enzyme. So once the hormone bind to the receptor, so there is a conformational changes, especially phosphorylation reaction happen in the tyrosine amino acid residue or this protein structure, and finally, you know, they will exert the physiological function, cellular response. So you can see that ion channel link receptor allow the ions in and out. Okay, that is a G protein link receptor. So the ligand, red colors refer to your hormones, ligand binding, activate the G protein, activate the G protein. So subunit of the G protein will bind to the hormone enzymes. In here, you will find the cyclic adenylyl cyclase enzyme, and activated adenylyl cyclase will release the cyclic AMP, second messenger swap. So enzyme link receptor, you can see that it's an inactive state. It's like that, no reaction occur at the other subunit of the receptor. So once hormone binding will allow the catalytic reactions, especially phosphorylation reactions occur, and later carry on the intracellular reactions and to complete their desired physiological function. Okay, so intracellular receptor, they are the very large superfamily hydrophobic signal molecule bind to the intracellular receptors. Okay, steroid and thyroid hormone have this kind of receptors, intracellular receptor. So it's allow the lipid soluble hormone to bind in it, and later they transported a hormone receptor complex. The combination of hormones and intracellular receptor is carried toward the DNA functional region, and later they will initiate the transcription to complete the protein synthesis. So protein synthesis means it's a intracellular, secondary intracellular response. So they have different domains, separate domain. First, that allow the hormone to bind in it. So DNA binding domain and transcription activation domain. So ligand binding domain and DNA binding domain are separated by the hinged region. Let's see the picture here in the next slide here. So this is a receptor structure. So receptor, of course, they are protein in nature. They have N-terminus part and then the carboxy terminus part, C-terminus. So the before hormone binding, there is an inhibitory protein complex bind to the DNA binding region, and they do not have any function yet. So you can see that this area called hinge region. So once steroid hormone come, they remove that inhibitory protein complex are detached from the binding. So hormone will come and bind, especially at the hormone binding site here. So DNA binding site exposed. So this exposed site will go to the functional gene in the, they pass through the nuclear membrane, of course, first, and later on they will go to the functional region. So they will bind to the gene. For example, cortisol receptor. In the cortisol receptor, their binding, DNA binding domain is a little bit towards the, close to the carboxy terminus part of the receptor. But estrogen different, you know, the DNA binding region, it's a little bit close to the N-terminus part. Okay, this is progesterone receptor. So when you check the receptor or the vitamin D receptor, you can see DNA binding region is at the N-terminal part of the receptor. Okay, thyroid hormone receptor, you can see in green color here. So retinoic acid receptor is also here. So they are the different DNA binding site belongs to the different types of the steroid hormones. Okay, so mechanism of group 1 hormone, shall we go? Okay, so as I mentioned earlier, hormone receptor complex is very important. So in order for the steroid hormone to show their action, so steroid hormone are secreted out from the adrenal cortex. Later on, they are transported by the globulin we call transcortin. Transcortin carry them until they reach to the target tissue. In the target tissue, transcortin deload its steroid hormone, and then steroid hormone can cross the cell membrane. When it reaches to the water median in the cytosol, they will bind to the intracellular receptor and form the hormone receptor complex. So hormone receptor complex will bind to the hormone DNA region we call hormone response element. And later, they will function to transcribe and translate. Okay, hormone response element is a part of the regulated expression region of the gene, behave like enhancer or silencer. So it's the site to initiate the transcription, that means enhancer. Sometimes it can inhibit the certain gene, so that means the action is to stop that particular gene activity. So in that case, they can call the silencer. Okay, okay, here you can see that. Okay, steroid hormone cross the cell membrane, later bind to the specific receptor, hormone receptor complex, HRC. We enter into the nucleus and bind to the specific DNA region and initiate the transcription. As a result, finally, protein is formed after the translation process. That protein play important role in the to show the physiological process. So these are the groups of hormones that use this what to call the intracellular receptors. Okay, okay, now it's the time to talk about the protein hormones and their mechanism of action. So they must require the second messenger to generate their function inside the target tissue. So they do not require transport protein. So it depends on the different classes of second messenger. And this group 2 hormone has different classes, class A, B, C, and D. Okay, class A, group 2A. So second messenger is cyclic AMP. How cyclic AMP is formed? Cyclic AMP is derived from the ATP actually by the enzyme adenylate cyclase. So intracellular level or cyclic AMP level may be high and decrease depend on the hormone, what type of hormone stimulated. Okay, so it varies from the tissue to tissue. For example, let's say cyclic AMP level may be variable in the heart muscles and then the skeletal muscles, so on. Depends on the state of their concentration. So interaction between the types of hormone and cell membrane receptor causes activation of membrane-bound trimeric GTP, so that bind the regulatory protein G protein. I think you have already got this kind of similar lecture in physiology, right? Physiology or pharmacology, my dear sisters and brothers.
Yes, doctor.
Hello.
Yes, doctor.
Yes, okay. So is consider is a revision, okay, my dear sister. So binding G protein, we will have a lot of, you know, the family GS, G stimulating, GI, GQ, G12, something like that, a lot. So G protein is actually trimerase protein, right? They are composed with these three subunit, alpha, beta, gamma. So alpha subunit bind to the GDP. Why we call G protein? So the protein is tightly bounded with the GDP, guanosine diphosphate. So in what you call that, GDP bound either alpha S or alpha I. Okay, inactive state is a GDP state. Once a ligand come, the receptor activate the G protein, alpha S. Alpha subunit will dissociate from the beta. Alpha subunit will bind to the adenylate cyclase and later activate the cyclic AMP. Okay, so the GTP action is what you call the GTP. In order to reform back to the inactive form, I think GTPase enzyme is required. GTPase degraded the GTP and will return to the GTP. So GTP, since GTP bind to the G protein, that is considered inactive form. Okay, okay, here you have already know. It's one of the example how the cyclic AMP play important role in representing the hormone to show the function inside. Okay, epinephrine and maybe other hormone. Okay, bind to the receptor. A G protein become active. Adenylate cyclase and then degrade the ATP and cyclic AMP release. So cyclic AMP will bind to the inactive form of the protein kinase. Actually, the protein kinase A made up with the 4 subunit, 2 identical catalytic unit and 2 identical regulatory unit. So cyclic AMP will make the protein kinase active. What is the active form? Active form means 2 catalytic units are detached from the 2 regulatory units, 1 cyclic AMP bind. So that the cyclic AMP binding regulatory unit will activate again to the phosphorylase kinase in active form. Phosphorylase kinase. Kinase means making the phosphorylation reaction. Okay, so the donor of the phosphate is ATP, of course. So ATP donate the phosphate to the hydroxy group, tyrosine amino acid residue of the phosphorylase kinase enzyme. So kinase enzyme become active. Phosphorylated form of the kinase is activated form. So active form will again to the activate to the other effector proteins. So that is in case of glycogenolysis pathway, phosphorylase B inactive form become active after it is activated by the phosphorylase kinase enzyme. Phosphorylase kinase enzyme make the attachment of the phosphate group to the inactive form and make the phosphorylase enzyme become active. So activated enzyme degrade the glycogen long chain to the glucose. So that reaction is physiological function found in our metabolism. That reaction is called glycogenolysis. So glycogenolysis is done by the epinephrine via this kind of series of reaction, finally glucose is formed, okay, from the glycogen. Okay. Okay, again, you can see that. And apart from this function, cyclic AMP has other functions. So it says activated protein kinase transported to the nuclear membrane. When it reached to the nucleus level, nucleus, intranucleus site, what happened is that it can activate the innate cyclic AMP response element binding protein. So make this protein activated. So activated protein can bind to the cyclic AMP response element binding element of the specific gene and activate the gene. So gene will start to transcribe and translate, and finally new proteins are formed. In case of glycogen phosphorylase, so enzyme synthesis is also can be regulated by this pathway in the gene, okay, at the functional gene level. So again, repeatedly showing this is how glucagon show, because glucagon also utilize the cyclic AMP as second messenger. So that's why here the regulatory, the important protein enzyme is a protein kinase A. Okay, show the phosphorylation to the other enzyme and show the action. For example, action of glucagon in the metabolism is glycogenolysis in liver. So the glucagon via this cyclic AMP, it's activate the glycogen phosphorylase, liver glycogen phosphorylase. So glycogen phosphorylase degrade the glycogen stored in the liver to the glucose. So this kind of reaction especially occur in the fasting time when blood glucose level low, right? So you can see that how cyclic AMP activated, first of all, protein kinase A activation occur, and then phosphorylase kinase A activation, later phosphorylase activation. So activated phosphorylase proceeds to complete the glycogen breakdown. Okay, so the cyclic AMP is, this slide's already explained. So for the hydroxylation reaction, occur especially at the serine or threonine or tyrosine amino acid residue of the phosphorylase and phosphorylase kinase. Okay, so these are the hormones that play important role in this group. They are second messenger cyclic AMP. So, but as I mentioned earlier slides, I told you, right, there's some of the hormone that stimulate the adenylyl cyclase to show their action. Some of the hormone that can inhibit the adenylyl cyclase. So these are the group of hormones, okay? So cyclic AMP. So because of cyclic AMP activation, and cyclic AMP carries the hormones action. So steroidogenesis, steroid hormone synthesis occur in the adrenal cortex by the ACTH. ACTH utilizes the cyclic AMP to synthesize the steroid hormone. This is we are try to correlate with the physiological function. And secretion of the iron, secretion of the hormones also depend on the iron transport. Sometimes use up the cyclic AMP, the carbohydrate metabolism, fat metabolism, activation of enzyme or enzymatic modification. We utilize the cyclic AMP. Enzyme induction via the cyclic AMP response element binding protein, gene regulation, cell growth and replication. So effect of cyclic AMP is very important. And then so cyclic AMP level can be decreased when enzyme is active. Activated phosphodiesterase will degrade the cyclic AMP, and cyclic AMP, once it's converts to the 5' AMP, their intracellular level will decrease. So sometimes cyclic AMP level, intracellular cyclic AMP level is higher, and sometimes cyclic AMP level is decreased when its activity is inhibited by the phosphodiesterase. So in a cyclic AMP, there will be the two parallel systems like stimulatory systems and then the inhibitory systems. So inhibitory hormones will act their action through the inhibitory systems and stimulatory systems. So stimulatory systems, as I mentioned earlier, GTP alpha S will activate the adenylyl cyclase and release the cyclic AMP. So cyclic AMP activate the kinase A and also phosphorylate the protein and show the cellular response. So how about the inhibitory systems? So inhibitory system has their own G protein. So there is a G inhibitory protein, also have 3 subunit like RS system, GS system. So they have alpha, beta, gamma, but inhibitory. So once they become active by the binding of the inhibitory hormone, so that way also activated form will bind to the adenylyl cyclase. So adenylyl cyclase, okay. So it's it will be inhibit the activity cyclic AMP, because it increases the phosphodiesterase and then inhibit the intracellular concentration of cyclic AMP level. If it decrease the intracellular level of cyclic AMP, okay. So there is a parallel system. Okay, see how? So inhibitory GI system. Okay, so alpha I inhibit the adenylyl cyclase and cyclic AMP. So beta gamma inhibit the subunit also adenylyl cyclase by binding to the adenylyl cyclase, and beta gamma also can bind to free alpha S, so preventing the activation of adenylyl cyclase. So you can see that your GI system, inhibitory system, has 3 action. Okay, inhibitory system inhibit the adenylyl cyclase enzyme directly. Okay, the rest part of the GI subunit beta gamma inhibit the adenylyl cyclase by binding to the adenylyl cyclase enzyme directly, or it can bind to the free alpha S from the stimulatory system and then prevent the activation of AC, because you know once alpha S is free, I mean the stimulatory alpha subunit is free, it will stimulate the adenylyl cyclase. Now it's no longer free by the binding of beta gamma. It will prevent the formation of adenylyl cyclase and then also inhibit the formation of cyclic AMP. Of course, cyclic AMP will decrease. Okay, got it? Do not confuse. Okay, so binding of adrenaline, example on the beta adrenergic, activate the G-alpha S, so increase the cyclic AMP level, intracellular cyclic AMP level in the target tissues like muscle. Okay, binding of adrenaline, but in separate receptor, alpha-2 adrenergic receptor will activate the GI. So that binding inhibit the adenylyl cyclase, so that's why it will decrease the cyclic AMP level. Okay, got it? Okay, got it? Got it? Okay, so here you can see that. So adrenaline or adrenaline in terms of what you call that, the function here, alpha-2 GI, you can see that the cellular action is there, adenylyl cyclase is inhibited, but here beta adenylyl cyclase is activated, so cyclic AMP increase, and then also what you call that, heart muscle contraction and smooth muscle relaxation occur. So as I mentioned earlier in the previous slides, the glycogenolysis, how adrenaline perform glycogenolysis is via the cyclic AMP response. Okay, cyclic AMP inhibited, but alpha 2 has two action. It also inhibited the phospholipase enzyme activity. So the inhibition occur, transmitter release is inhibited. And the one inhibition cause smooth muscle contraction. So other type, alpha-1 receptor, even same adrenaline, when it's bind to the alpha-1, it will work in the other G protein and also stimulate the phospholipase C. That is a group 3 enzymes. It can be found in the group 2C enzyme hormones activity. I show you in the next slide. Okay, IP3 become active. As a result, calcium is formed. Calcium will bind to the chemogulin complex and form calcium chemogulin complex that will activate the contractile muscle like myosin, so they cause smooth muscle contraction. So giving an example. Okay, so resting state, you can see the epinephrine receptor do not react with the G protein and adenylyl cyclase. Once hormone binding will activate to the protein one by one. So finally, so it will, you will receive the physiological response like increased heart rate, vessel dilatation, glycogenolysis, so on and so forth, right? My dear sisters and brothers. So, okay, so I would like to share some biomedical importance here regarding the cholera toxin and whooping cough pertussis. Okay, so cholera toxin, cholera is diarrhea, severe diarrhea, loose motion caused by the cholera vibrio organisms, right? You have already know. So what happened is that the cholera toxin here, G-alpha S is ATP ribosylation. That means attachment of ATP ribose sugar to the G-alpha S. So G-alpha S maintain the active state. GFRS is the active state, right? So keep activating the adenylyl cyclase. So keep increasing the cyclic AMP level, keep increasing the protein kinase A. So in that case, the result is that, you know, enhance the cyclic AMP production, causes the opening of the chloride channel. So chloride is lost. So hypochloremia occur. And in sodium, potassium, hydrogen ion exchanger. So as a result, the chloride efflux occur, and also water is low, no water absorption. Finally, you know, they will present with the loose motion and then the electrolyte imbalance. It is a kind of fatal disease, unless they receive, the patient receive the adequate amount of fluid replacement like normal saline and ring galactic saline and dextrose saline. So because there are, you know, the several times of loose motion can cause the hypovolumic shock and fatal situation. So because of the changes happen in the intracellular changes happen by the our cholera toxin. Okay, gram-negative bacteria. Okay, so pertussis, whooping cough. Whooping cough is caused by pertussis organisms, right? So we have immunization program since baby is born, and then they have to inject with the inoculate with the that vaccination. Okay, pertussis vaccine. So it's also kind of the fatal. So the mechanism of action, how the toxin written is that they activate, the toxin activate the ATP ribosylation reaction to the GI alpha. So GI alpha, so ATP ribose sugar, once it attached to the GI alpha, GTP, so GI alpha, GTP is a key, what to call that, active. So the activation will inhibit the adenylate cyclase. No cyclic AMP is formed. So the result is that complaints remain bound to the GTP inactive state. So unable to inhibit the adenylate cyclase, that condition open the potassium channel. So as a result, the person, the bitten will complain and will present with the productive cough, non-stop cough, and then it will lead to respiratory failure. So this is how the toxin work in the our G alpha S system and G alpha I systems. Okay, just sharing. Biomedical important. So now it's a time to talk about the second messenger cyclase GMP. So the receptor bind to the guanylate cyclase directly. So they do not need the G protein in that case. So the GTP is catalyzed by the guanylate cyclase, form cyclic GMP. There will be the two form of guanylate cyclase, soluble form or membranous form. Membranous bound to bind to the membrane. A soluble form will be present, available in the intracellular space. So ligand binding, so cyclase active, direct rather than the G protein, right? So the hormones that use the cyclic GMP as a second messenger are atrial natriuretic peptide, which are available in our atrium. So membrane bound form, okay? Target organ are kidney, blood vessels, and adrenal cortex. Effect is the excretion of sodium. So removal of the sodium, extra sodium. So once sodium is excreted from the renal tubules, and water also follows. And so diuresis and natriuresis is as a result of hormone actions, okay? On the blood vessel, it's called the cause vasodilation, relaxation of smooth muscle, and it also inhibits the aldosterone, so that aldosterone function is salt and water retention, right? So it will be inhibited the aldosterone function, so it causes the diuresis and natriuresis. So the hormone action is that you can see here. Atrial natriuretic peptide or natriuretic factor. So it is released not only from the atrial tissue, but also can be synthesized in the intestinal lining, blood vessel, brain, and kidney in small amount. So once the hormone bind to the receptor, guanylate cyclase enzyme is a receptor as well. So guanylate cyclase will activate the, catalyze the or hydrolyze the GTP and form cyclic GMP. So once cyclic GMP is high, so cyclic GMP will activate its relevant protein kinase. That kinase, protein kinase G. We call protein kinase G. It's different from A. A is activated by the cyclic AMP. Okay, so that protein kinase will, what you call that? Phosphorylate the smooth muscle protein. So as a result, we cause the smooth muscle relaxation, vasodilation, because the myosin light chain become relaxed. And then also it causes the diuresis, natriuresis in the kidney, and also inhibit the aldosterone secretions in the adrenal cortex. So there are some other drugs that also have activities to bind the guanylate cyclase is nitroprusside, nitroglycerin. So that drugs is used. You have already studied about the nitroglycerin, right, in your cardiovascular system. So that is used the ischemic, anti-ischemic agent, you know. Okay. Okay, my dear sisters, brothers. Okay. So effect is smooth muscle relaxation resulting vasodilation. Let the cardiac muscle receive the more blood supply. Okay, so cyclic AMP is degraded by phosphodieters. So cyclic GMP level may be lower once it's done the function. So by the enzyme phosphodiesterol will degrade the phosphate and the cyclic AMP will convert to the 5' GMP. So action will stop. So inhibitor of cyclic GMP phosphodiesterase. For example, let's say that enzyme is inhibited. Could you imagine that your cyclic GMP level will be keep increasing and maintain high in the cell, right? So enhance the prolong the response, smooth muscle relaxation, vasodilation. So it is this action is become drugs. Have you ever heard about this drug, Viagra, sildenafil? So sildenafil is increase the libido, blah, 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 blah. So increase the cyclic GMP level in the smooth muscle, especially in the sorry to say, because we are health professional, we have to explain about the this and that, okay? If I say wrong, please forgive me. Okay, so cyclic GMP level, increase the cyclic GMP level in testicular muscles. So as a result, what happened is that phosphodiesterase level is inhibited. So cyclic AMP level is high. So as a result, so it increase the corpus cavernosum cyclic GMP level, increase the smooth muscle relaxation. So let the blood come into the corpus cavernosum. So they receive their desire, what you call that function. So sildenafil is a recommended doses, has no effect. Okay, in the absence of sexual stimulation, blah, blah. It's okay. So what I want to highlight is the mechanism of action of this drug. Because the pharmacologists and scientists, once they realize that if they inhibit, able to inhibit the phosphodiesterase, so they can keep the high concentration of cyclic GMP. Okay. Okay, let me continue other. So I want to talk about the cyclic AMP as the group 2C hormone. The second messengers here are calcium and then phosphoinositol and diacylglycerol. So there are groups of hormone that higher the cyclic, what you call the group 2C second messenger. So here is a hormone binding on the hormone with bind to the receptor, activate the GQ. So activate the GQ will stimulate the phospholipase C beta. So lipase C beta will hydrolyze the phosphoinositol diphosphate. Actually is a component of cell membrane. So it will produces the two messengers, second messenger, inositol, triphosphate, and diacylglycerol. You can see here, my dear sisters and brothers. So phospholipase C is activated. So it's a degraded phosphoinositol diphosphate to IP3 and DAG. So IP3 will activate the what you call that endoplasmic reticulum and intracellular mitochondria to release the calcium. So calcium is leaked out from the subcellular organelles. So make your cytosol increase calcium level inside the cytoplasm. So once calcium level is high, calcium will activate the protein kinase C. So become active. Diacylglycerol is also similar ways, the different way that can activate the phosphokinase C activity. So as a result, the phosphokinase C activated form will activate to the other enzymes and substrates. So in order to show their physiological function. So at the same time, the calcium is also considered the second messenger. So what happened is that the IP3 also can activate not only to activate the subcellular organelle, but also can activate the calcium channel. So let the calcium channel open. So calcium extracellular calcium will enter into the cell, and finally that will increase the intracellular calcium level and keep activating the protein kinase C to complete the physiological function. The same picture here, so physiological function. So how about the calcium? So when you look at this picture, so calcium will bind to the specific protein called calcium binding protein or chemogulin. So chemogulin has many functions. Not only activating the protein, so they have, special activities. So they can activate the other protein. So there are multifunctional chemogulin kinase also can activate the various types of the intracellular proteins to show the physiological functions. Okay, my dear sisters and brothers. So this is how gonadotrophin releasing hormone, thyrotrophin releasing hormone activated. So bind to the receptor, GQ protein is activated. So we'll what to call that IP2 inositide diphosphate is degraded. So phosphodinositide diphosphate is catalyzed by the protein kinase C. So IP3 is released. So IP3 can be recycled also. So as a result, you can see that it exerts the physiological function. So it is already explained. Okay, so chemogulin kinase is also called CAM kinase. Okay, IP3 is actually short-lived chemical messengers. Most IP3 is dephosphorylated by the phosphatase and found, and then also some of the IP3 can be phosphorylated and added by additional phosphate and form IP3. They can be recycled. Okay, so the enzymes, okay, glycogen synthase, pyruvate kinase. Glycogen synthase is an enzyme that involves in glycogen synthesis. Pyruvate kinase enzyme and dehydrogenase and carboxylase enzyme. These enzyme activities are found in your Krebs citric acid cycle. They are activated by the calcium activated phosphorylation process. Okay, and then last but not the least, so these are the enzyme-linked cell surface receptors or transmembrane tyrosine specific protein kinase. They are the single pass transmembrane receptors and with the large glycosylated extracellular protein that allow the epidermal growth factor to bind in it. So let's see how the receptor. So here, let me show you the picture first. So the growth factor bind to the tyrosine kinase receptor. So the intracellular part of the subunit of the receptor undergo the phosphorylation process, especially in the amino acid, tyrosine, serine, threonine, and we found the rest protein. And then later, they will bind to the other kinase protein, and that protein is able to activate the functional gene and transcribe, and proteins are formed. So in case of, you can see that tyrosine kinase receptor utilized by the growth factor, cytokine utilize the the serine and threonine kinase receptor. Here you can see that. Okay, so I would like to highlight a little bit detail about the insulin. You know how insulin is formed, right? Insulin require the external stimuli. Extra glucose concentration is higher after glucose enter freely to the beta cells because glucose transporter 2 doesn't need the action of insulin to allow the glucose to enter the cell. So you can see that glucose enter freely. Once glucose enter into the cell, in the cytoplasm, glycolysis reaction occur, provide the ATP, and then also glucose convert the pyruvate at the end of the glycolysis. So the pyruvate will enter into the mitochondria and proceed the Krebs citric acid cycle. As a result, a large amount of ATP is formed. So ATP is formed extracellular ATP is formed in higher amount. ATP will close the potassium channel that cause the intracellular potassium levels high, that cause the depolarization, that causes the opening of the calcium bonded channel, let the calcium enter. So calcium, see how calcium is important for the release of insulin from the beta cell. So push the vesicle, insulin vesicle towards the membrane later. The insulin is secreted and transported to the tissues, target tissue. What happened in the muscle, target tissue or insulin? So insulin here bind to the receptor, alpha subunit. So beta subunit, tyrosine phosphorylation occur, and also again activate to the other series of enzyme to become active, and then so the activation, one of the activation of enzyme, so the enzyme can be transported into the gene, functional gene inside the nucleus. Isn't it functional gene? So activate the what you call the functional gene of the glucose transporter. So make the glucose transporter 4 synthesize more and more. So glucose transporter 4 will present in the cell membrane and allow the glucose to enter into the cell. This is how insulin activate the glucose transporter 4 in the muscles, adipose tissue, and other tissue. So these tissue require the insulin to receive the glucose. So later on, so we say that insulin is hypoglycemic hormone that lower the blood glucose level. This is the mechanisms how insulin lower the blood glucose level, because that open the all the glucose transporter 4, so the extracellular glucose can enter the cell and lower the plasma calcium level. Apart from this function, insulin has some other many important functions. For example, it increases the growth factors, protein. It also involved in the tissue growth. It also involved in the lipid metabolism, protein metabolism, nucleoprotein metabolism. There are so many factors insulin involved, right? Shall we go back to that slide? I think I already explained it. So in that group, you can see that the tyrosine phosphorylation can stop when the phosphate group is removed from it. So that means the receptor remain in inactive state when the fasting state, when insulin or ligand does not come and bind it, right? Okay, these are the factors, growth factor. They utilize this protein kinase cascade for them to show their function inside the target tissue. Okay, I think it's done. So I summarize that a brief action of cell signaling through the gene expression, through the enzymatic activity, through the changes, series of reactions happen inside, including the electrolytes and membranous potential. So these are the hormone response, metabolic action, cell growth and differentiation, cell motility and replication, transcription, translation, granule release or hormone release, endocytosis, so on and so forth. So this slide is a summary of our what you call the lectures. So ion transport, you can see there, membrane receptor, activate the G protein, also activate the tyrosine kinase, alter the cytoskeleton. As a result, you can see that alter the protein, show the cellular response. Cellular response includes the enzyme induction, enzymatic activation, activation of the multi-protein like myosin, and then membrane receptor synthesis like glucose transporter synthesis, and gene activity and other types of protein synthesis. So they all work together, what you call that, systematically, very good, working, we result the good function and we maintain our normal function. So this is a clinical application I think you have already known. And then beta blockers that used in treatment of hypertension, and then the calcium channel blocker, they block the receptors and ion channel respectively, okay? And also H1 and H2 receptor blockers also work at the site of the receptor. So estrogen receptor blocker named tamoxifen is used to treat the estrogen dependent breast cancer. And angiotensin receptor blocker is used as a hypertension, used for the antihypertensive agents. So protein kinase inhibitors may be the effective anti-cancer drugs nowadays. So just sharing the caring, any information, any questions? My dear sisters and brothers, that is all about my lecture. Okay. 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 Questions? Christina? Brother Ahmed Akbar? Shamsuri? Brother Shamsuri? Brother Nabil? Are you here? Brother Arif? Any questions? Sister Haja? Any question? Are you here? Are you available?
Yes, doctor.
Okay, okay. No
Yes,
response.
doctor.
Okay. So any questions so far? A little bit fast because I know that please consider that this is a revision for you to study the further lectures, you know, very important. Especially for the biosynthesis, especially in the pharmacology. So you will be learning a lot of drugs like oral contraceptive drugs will be delivered by our Dr. Raza. There are a lot of steroids hormone functions there, you know, steroid hormones, how to use the steroid. And you will be also studying about the medicinal chemistry or drugs, detail of steroid drugs, and then oral hypoglycemic agents, insulin, so on and so forth later. Okay, Sister Shafika, any questions so far? Sister, Brother Hasnu, Sister Atifa, stay awake. Okay, no question, clear, right? Okay then, so I think it's a time to say bye and see you again. In next lecture, I will have altogether 6-hour lectures, including tutorial. Okay. Okay, one question. Yes, Christina, any questions? You can switch on your microphone so that everybody can hear. Christina, Sister Christina. Hello. Oh, microphone has problem. Okay, okay, you can type your questions. I will try to answer. What's your question? All clear. Is it clear, Brother Muhammad Siddiq? Brother Shami Fikri? But I think it's very simple lectures, and then later on you will be studying the more difficult part. Group 2C, okay, thank you. No problem. No problem, I can repeat. Very simple. Okay, Group 2C. Group 2C, okay, the hormones binding, can you hear? Hormone binding will activate the receptor. Receptor will activate the specific G protein. That protein is GQ protein. So protein, also the similar way, this G protein composed with the 3 subunit, G alpha, beta, and then the gamma. So G alpha will activate the protein kinase C. So action of protein kinase C degrade the phosphoinositol diphosphate phosphoinositol, PIP2, phosphoinositol diphosphate. That is a membrane component. Your membrane is phospholipid, right? Phosphoinositol diphosphate are degraded and to the what you call the IP3 inositol triphosphate and diacylglycerol. Phospholipase C, and diacylglycerol and IP3. So diacylglycerol and IP3, the action is there. Diacylglycerol can directly activate the protein kinase C. So activation of protein kinase C will activate the what you call that effector proteins that will involve in physiological function. Phospholipase C action, phospholipase C degrade the PIP2 into 2 molecules, right? How about the action of IP3? So IP3 activate all the subcellular organelle to release the calcium. So calcium is leaked out from the subcellular organelles such as the endoplasmic reticulum and then the mitochondria. Finally, what happened is there, intracellular calcium level is increased. And not only there, IP3 also can increase, can activate the ion channel, calcium channel. So let the calcium enter to the cell, from the outside, extracellular fluid. So you have excessive amount of calcium. So calcium will bind to the, this is chemogulin. So chemogulin complex, calcium chemogulin complex will have to have many functions, cellular function. Some of the chemogulin complex activate the some storage granules to move towards the cell membrane. So that reaction only happen in pituitary glands, for example, pituitary hormone. So the storage granules are pushed towards the membrane. Later, pituitary hormones are excreted out. So that means that hormone like thyrotrophin releasing hormone, right? Gonadotrophin releasing hormone, they are synthesized in hypothalamus. Okay, these are synthesized in hypothalamus when they bind to the receptor that are present in the anterior part of the pituitary. So their action is that the secretion of the pituitary hormones. For example, TRH. TRH function is the stimulation of the thyroid stimulating hormone. So the calcium chemogulin complex push the vesicle towards the membrane and exocytosis reaction occur and thyroid stimulating hormone is released out from the anterior part of pituitary. You can imagine that this thyroid stimulating hormone is transported to the thyroid glands later on, whereby thyroid glands secrete the T3 and T4. Okay, Christina, have you got it? Have you got it? Hello. Hello. Other friends, have you got it? Other sisters and brothers?
Very.
Okay. Okay. Alia, thank you. You got it. There's a point in the slide, GI. Okay, Christina. BY. BY, is it? Okay. Beta gamma subunit are the same. Alpha S. Which slide is it? Alpha I. The different. What is it? Okay. Let me go there. Which slides you refer to? Which slide? Is it this slide? Which slide? Which slide? Is it this slide?
The slide on inhibitory
this
G protein.
Oh, this slide, is it? Is it?
Yes.
Okay. Okay, this slide. Okay. Okay, GI and GS system is a different system, okay? So because G protein has a specific system, inhibitory system and stimulatory systems, you know. Okay, so stimulatory system is also walk by the specific hormones, whereas inhibitory system will be also influenced by the other different types of hormone. Okay, so when you check the picture here, sister, here, we have receptor GS system and GI system, right? Okay, so there are two different systems here, right? This slide, is it? Which slide? I confuse again. Here, this slide, is it? Okay, beta gamma, okay? So both alpha 1, also inhibitory G protein also contain beta gamma subunit and alpha subunit like G stimulating protein. So they have beta gamma subunit, so they act as an inhibitor. Okay, for the GI subunit, alpha I inhibit the adenylyl cyclase. By inhibiting adenylyl cyclase, it inhibit the cyclic AMP formation. Okay. How about other beta gamma action? Beta gamma also inhibit the adenylyl cyclase by binding to the adenylyl cyclase, and also they have an activity to bind the alpha stimulatory unit, alpha S. Okay, alpha S derived from the GS system. Got it? So beta gamma from alpha, what to call that, GI protein, GI inhibitory protein has beta gamma subunit. So that subunit binds to the alpha S, and so that alpha S cannot bind to the adenylyl cyclase. Because you know why? Adenylyl cyclase activation depend on the binding of G alpha S. Now G alpha S cannot bind to the adenylyl cyclase. There is no action of adenylyl cyclase, so there is no cyclic AMP is formed. So this is how GI subunit, GI inhibitory system inhibit to the adenylyl cyclase. Okay. So beta gamma subunit are same in GS and GI, but alpha S and alpha I are different. Yes, of course, because what I want to say in terms of structure, they are the same, beta gamma, okay? GS and G alpha I, but G alpha S and alpha I subunits are totally different in terms of structure. For example, G alpha S also has subunit alpha. Alpha S in terms of structure is different from the alpha I. Okay, got it? Got it, Christina? Sister Christina, other friends? Hello.
Yes, Doctor.
Is it?
Get it.
Yes. Is it? Okay. Oh, sorry. What happened to your... Okay, everybody hear my lecture? The whole lecture?
Yes, Doctor.
Yes, sir.
Okay, thank you, Sister Atika. How about Brother Samsuri? No sound from you? Brother Arif, are you a class rep? Is it? Or PIC? Who's a PIC in your class for this subject?
I am the PIC.
I didn't hear you. Okay. So I think it's a time to stop. I want to stop earlier, but it takes time. Okay then. So I think let me stop here. Okay. Bye-bye. See you again. Thank you.
Thank you very much, Doctor.
Thank you so much, Tutu.
Thank you, Doctor.
You're welcome.
Thank you, Doctor.
So let me leave. You can leave, eh? You can leave freely. Okay.
Thank you very much.
Bye. But if you do not understand and realize my lecture, you can come to me again anytime. Okay, WhatsApp me, Telegram me. Okay, I'll be always there. Okay, bye bye.